After studying this module, you will be able to know about pharmacodynamics of drugs, mechanism of drug metabolism, and identification of the receptor or enzyme which helps in drug action. First, introduction to pharmacodynamics. Pharmacodynamics is the study of biochemical and physiological effect of drug and their mechanism of action at organ level as well as cellular level, or it may be referred to as the association between drug concentration at the site of action and subsequent effect comprising the time and intensity of therapeutic and unfavorable effects. The effect of a drug existing at the place of stroke is determined by the drug's binding with the receptor. Receptors may be present on neurons in the central nervous system, example in case of opiate receptors, to depress pain sensation on cardiac muscle to affect the intensity of contraction, etc. For most drugs, the concentration at the site of the receptor determine the concentration of a drug's effect. Next is the principle of drug action. Drugs do not demonstrate latest functions on any system, organ or cell. It only alters the pace of ongoing activity by different actions. First, the principle, mode of action and the example. Principle is stimulation. Mode of action, selective enhancement of level of activity of specialized cells. Excessive stimulation is often followed by depression of that function. Example, pilocarpine stimulates salivary glands. Picrotoxin, CNS stimulant, gives rise to convulsions, which ultimately gives rise to coma and leads to the death. Second principle is depression. In this, the mode of action is selective diminution of specialized cells. Certain drugs stimulate one cell type and depress other. Example, barbiturates depress central nervous system. Quinidine depresses heart. Ache stimulates smooth muscles but depresses SA node. Next principle is irritation. Mode of action is non-selective, often noxious effect applied to less specialized cells like epithelium or selective tissue. They stimulate associated function. Example, bitter salivary and gastric secretion counter irritants increase blood flow to a site. Next principle is replacement. Mode of action, use of natural metabolites, hormones or their congeners in deficiency states. Example, levodopa in Parkinsonism and iron in anemia. Next principle is cytotoxic action. Mode of action, selective cytotoxic action for invading parasites or cancer cells for attenuating them without affecting the host cells. Example, penicillin and chloroquine. Next is the mechanism of drug action. The drug action depends on the biomolecules, typically a protein. First, enzymes. All biological responses are taken out in catalytic influence of enzymes. Drugs have capability to upsurge or reduce enzyme-mediated reactions. The enzyme stimulation is not common in drugs or generally by endogenous substrates. Enzymes such as acetylcholine esterase, choline acetyltransferase, cyclooxygenase, xanthine oxidase, angiotensin converting enzymes, carbonic anhydrase, HMG-CoA reductase, dopa carboxylase, monoamine oxidase, dihydrofolate reductase and DNA polymerase. Enzyme inhibition. Common modes of drug action. Non-specific inhibition, denaturation of proteins, strong acids, heavy metals, alkalis, alcohol, phenol, etc. Then specific inhibition, They are divided as per the effect of fluctuating intensity of the enzyme's substrate on the inhibitor. In competitive inhibition, the substrate and inhibitor will not bind to the enzyme at the same time. This generally consequences from the inhibitor having an affinity for the active site of an enzyme, where the substrate also binds. The substrate and inhibitor compete for access to the enzyme's active site. This kind of inhibition can be resolved by appropriately high concentrations of substrate. Vmax remains constant, that is by outcompeting the inhibitor, though the apparent Km, that is the Michaelis constant, will upsurge as it attains a high concentration of the substrate to reach the Km point or half the Vmax. Competitive inhibitors are generally same in organization to the actual substrate. In mixed inhibition, the inhibitor can attach to the enzyme simultaneously as the enzyme's substrate, though the binding of the inhibitor interrupts the binding of the substrate and vice versa. This kind of inhibition can be decreased but cannot come across by amplifying concentrations of substrate. Even though it is probable for mixed type inhibitors to join in the active site, this kind of inhibition usually falls out from an allosteric effect where the inhibitor attached to various sites on an enzyme. Inhibitor binding to this allosteric site changes the conformation, that is, tertiary structure or three-dimensional shape of the enzyme, so that the affinity of the substrate for the active site is reduced. Non-competitive inhibition is a form of mixed inhibition, where the binding of the inhibitor to the enzyme reduces its activity but does not affect the binding of substrate. Due to this, the extent of inhibition depends only on the concentration of the inhibitor. Vmax will decrease because the reaction proceeds efficiently, but Km will remain the same as the actual binding of the substrate. By definition, will still function properly. Second is ion channel. Proteins take part in transmembrane signaling and regulate ionic composition. Ions like receptor or voltage gated sodium, potassium, calcium and chloride channels. Drugs also target these channels: ligand gate channels, G protein operated channels and direct action on channels. Third is transporters. Drugs interact with these transport systems in which substrates get translocated through the membrane when they bind to specific transporters, that is carrier. Solute carrier protein. They pump the metabolites or ions relative to the direction of concentration gradient or against it. Example: probenecid, penicillin and uric acid, furosemide, that is sodium, potassium, calcium and carbon monoxide transport. Hemicolinium, choline uptake and vesamicol active transport to ACH to vesicles. Fourth are the receptors. It is described as a macromolecule or binding site situated on cell surface or within the effector cell that act to distinguish the signal molecule or drug and initiates the response to it, but itself has no further function. Example: G protein coupled receptor. The word receptor was introduced in 1909 by Paul Ehrlich. Drugs generally do not bind directly with enzymes, channels, transporters or structural proteins, but act by particular macromolecules. John Langley has been credited with the theory of drug acting on receptors. While studying the antagonistic effects of atropine against pilocarpine-induced salivation, he observed there are several elements in the cells of gland and endings of nerve cells with which both atropine and pilocarpine are competent of forming compounds. Langley later stated these factors as receptive substance. A receptor can exist in at least two conformational states, active and inactive, that is RA and RI respectively. If these conditions are in equilibrium and the stationary condition predominates in the non-existence of drug, then the basal signal results will be low. The extent from where the equilibrium is fluctuating in the direction of the relative affinity of the drug for the two conformations. First, agonist, an agent that stimulates the receptor to create an effect analogous to that of the physiological signal molecule. Example, muscarine and nicotine. Next is antagonist, an agent that prevents the action of an agonist on a receptor or the succeeding reaction, but does not have an effort of its own. Example, atropine and muscarine. Next is inverse agonist, an agent which turns on receptors to produce an effect in the reverse direction to that of the agonist. Example, DMCM. Next is partial agonist, an agent which turns on a receptor to create submaximal effect but antagonizes the turn on of a full agonist. Example, pentazocine. Next is ligand, those molecules which attach selectively to specific receptors or sites only in the binding or affinity. Mentioned terms are explained in terms of affinity and intrinsic activity, which means affinity. It is a measure of propensity of a drug to bind receptor, the attractiveness of drug and receptor. Next is intrinsic activity or efficacy or IA. It is defined as the property of a bound drug to modify the receptor in such a way that creates an effect. Some drugs possess affinity but not efficacy. Then agonist is affinity plus IA1, antagonist is affinity plus IA0, partial agonist is affinity plus IA0 minus 1, and inverse agonist is affinity plus IA0 to minus 1. Next is drug receptor binding. D plus R, that is drug plus receptor affinity, gives a DR complex, that is drug receptor complex. DR complex gives effect, that is E. Covalent bonds are steady and fundamentally irreversible. Electrostatic bonds may be strong or weak but are typically reversible. Functioning of receptor. Two necessary roles of receptor are: first is recognition of specific ligand molecule, and second is transduction of signal into response. We can see the picture which shows all domains are ligand binding domain, and then effector's domain undergoes functional conformational change. Cell surface receptors floats in cell membrane lipids. Roles are determined by the relation of lyophilic or hydrophilic sphere of the peptide chain with the drug molecule. Nonpolar hydrophobic piece of the amino acid stay hidden in membrane, while polar hydrophilic stay on the cell surface. Hydrophilic drugs cannot cross the membrane and has to connect with the polar hydrophilic piece of the peptide chain. Binding of the polar drugs in ligand binding domain includes conformational changes, alter distribution of charges, and transmitted to coupling domain to be passed on to the effector's domain. Drugs have an effect on the physiological receptors and arbitrate reaction of corticoids, hormones, transmitters, and others such as cholinergic, adrenergic, or histaminergic, etc. Drugs may proceed on true drug receptors like benzodiazepine receptors. Next is transducer mechanism. The majority transmembrane signaling takes place by a minute quantity of diverse molecular process, that is transducer mechanism. Large number of receptors shares these handfuls of transducer mechanisms to produce a combined reaction. The four chief categories are GPCR, that is G protein coupled receptor, receptors with built-in ion channel, enzyme associated receptors, and transcription factors, receptors for gene expression. GPCR, that is G protein coupled receptor, large family of cell membrane receptors associated to the effectors, enzyme, channel or carrier proteins by more than 1 GTP turn on proteins, that is the G proteins. All receptors has common pattern of structure organization. The particle has 7 alpha helical membranes across hydrophobic amino acid segments, 3 extra and 3 intracellular loops. GPCR 3 major pathways. There are 3 pathways. First is adenyl cyclase CAMP pathway, phospholipase C, that is IP3 DAG pathway, and channel regulation. Stimulated G proteins will on or off ion channels, that is calcium, sodium or potassium, etc. These effects may be present with no interference of any two messengers, that is CAMP or IP-DAG. Brings about depolarization, hyperpolarization or calcium changes, etc. GS, calcium present in medical drug proceed by impersonating or blocking the actions of endogenous ligands that control the flow of ions by plasma membrane channels in myocardium and skeletal muscles. GO and GI opens potassium channel of muscles and heart and closes calcium channel of neurons. The natural ligands include acetylcholine, serotonin, aminobutyric acid, that is GABA, and the excretory amino acids, example glycine, aspartate and glutamate. Enzyme linked receptor. 2 types of receptors. First, intrinsic enzyme associated receptors, protein kinase or guanyl cyclase domain. Second is JAK STAT kinase binding receptor. Extracellular hormone has 2 binding domain and a cytoplasmic enzyme domain, mainly protein tyrosine kinase or serine kinase. On binding, the receptor changes from its inactive monomeric state to an active dimeric state. Cytoplasmic area turns out to be phosphorylated on particular tyrosine residues. Enzymatic activities are activated, catalyzing phosphorylation of substrate proteins, and then activated receptors catalyze phosphorylation of tyrosine residues on different target signaling proteins, thus permitting a single kind of turn-on receptors to adapt a number of biochemical procedures. Examples: insulin, intake of glucose and amino acids, and controls metabolism of glycogen and triglycerides. Trastuzumab, antagonist of such kind of receptor used in breast cancer. Receptor regulation gene expression, lipid-soluble biological indicator across the cell membrane and proceed on intracellular receptors. NO works by exciting CGMP. Receptors for corticosteroids, mineralocorticoids, thyroid hormones, sex hormones and vitamin D, etc. excite the transcription of genes in the nucleus by binding with specific DNA sequence called responsive elements. Hormones generate their effects after an interval period of 30 minutes to several hours. The time needed for the production of latest proteins gene-active hormonal drugs needs time to become active. Bronchial asthma, a favorable or toxic effect remains even after removal. We'll conclude this module with summary. The subdivision of pharmacology that relates drug concentration to biological effects is known as pharmacodynamics. Fundamental principle of pharmacology is that drugs must interact with the molecular target to exert an effect. Drug interaction with molecular targets is the initiating event in a multi-step process that ultimately alters tissue function. Agonists activate receptors to produce a response. Antagonists bind with receptors but do not activate them or cause a response. They can essentially block the activation of receptors. Partial agonists produce a response. However, this is less than would be expected by a full agonist drug. Inverse agonists are drugs which can reduce the normal activity of the cell. Competitive antagonists are drugs that prevent activation of the cell by the normal agent. Non-competitive antagonists are drugs that may block the receptor but not in a permanent way.